Well, then how about phenylalanine? Which carbon should be here? CH2. By the way, a good name for this carbon would be the beta carbon. If this is the alpha carbon, then this would be the beta carbon. And then this would be the rest of the side chain. You can see why this is called phenylalanine, by the way, because this is a phenyl group. And an alanine would be if we just had a methyl group here. So it's a combination of alanine and the phenyl group.
How about the proline?
Yeah, that's one of the trickier ones. In fact, that doesn't really follow the same basic pattern, so we have to learn that one separately. Here's our basic structure for an amino acid. And now the side chain here, we have a 3-carbon side chain. 1, 2, 3. Proline has a 3-carbon side chain, and the interesting thing about the side chain is that the end of the side chain loops around and connects to the N-terminus.
Oh, so they're giving us the N-terminus and the C-terminus in the R group for some reason?
Because proline is so different from the other amino acids, for proline, they drew the entire amino acid.
Okay.
The picture in the table there is the entire picture of proline. There's no real way to just draw the side chain for proline because the side chain is attached to this nitrogen here. So they've broken from their normal format here in the table. For proline, they actually drew the entire amino acid. What I have on the board here is just a copy of what's in the table. This is just the entire amino acid. This is pretty normal, except for this bond here. This one over here?
Oh, sorry.
This is just the normal carboxy end. This is just the normal carboxy end. I didn't draw it quite the way they have it there in the table, but what I have on the board is the same thing they have there. I tried to draw it so it looks a little bit more like the other drawings we've had of the amino acids so far. And again, the key thing is, this is a perfectly normal bond between the alpha carbon and the first carbon in the side chain. The only weird thing is that the end of the side chain is also bound to the N-terminus. This is the only amino acid where we have that. So by the way, the neutral form here the neutral form of the N-terminus usually has 2 hydrogens. But here the neutral form of the N-terminus has to have 1 hydrogen because it has an extra bond to a carbon.
Right, so can we do some what happens when we have pHs here?
Sure. So let's say that we were at a very low pH. Is there anybody I have to add a proton to? That's right. So this would be the form at a very low pH. How low does the pH have to be for that to be the major form?
Less than 9.6.
Right. Oh, well, in order for this to be the major form of the carboxy and the nitrogen, I guess it would have to be lower than 2.0 since I've got them both protonated. But in order to protonate the nitrogen, you're right, it just has to be lower than 10.6. That's right. Now, again, notice, normally when you have a protonated N-terminus, it has 3 hydrogens. But when proline is protonated, it only has 2 hydrogens because it has the extra bond to the carbon. So you always want to make sure that you're not breaking the octet rule. So this is what we would look like at a low pH. And how about if we were at a high pH? If we were at a very high pH. We would get rid of that proton and put the negative charge here.
So we have a +1 and a -1 charge.
That's right. And there was an intermediate case that I didn't draw where one of the things where they would both have charges. In any case, the key thing is we saw what does the N-terminus look like when it's protonated in proline? Well, it looks a little bit different from a normal N-terminus because it has 1 fewer hydrogen. The key thing to have in your notes here is that, in fact, so the key thing to have in your notes here is that the table is different for proline. For all the other amino acids, they only drew the side chains. But for proline, they drew the whole thing. In fact, if you look at the little figure there, notice how the little figure has a tiny little b?
Yeah.
And that tiny little b at the end is to remind you that's the entire structure.
Yes. So if the pH were like
For proline?
Mm-hmm.
Okay. Are you working that out?
Yeah.
Okay.
Is this right if pH is 1?
Now, would we expect everybody to be protonated here or deprotonated?
Well, wait. So 1 is less than the cutoff point for the
Right.
carbonyl.
Right.
So the carbonyl, that would be O minus. And then
Let's take our time there. Now, are there a lot of protons here or only a few protons?
3.
I think you might have gotten
Sorry,
confused.
a lot of protons.
A lot of protons. So should the functional groups here have lots of protons or few protons?
Oh, lots.
A lot. That means this is not the correct form of the carboxylate here. The carboxylate here would pick up a proton from the solution, and this would be the form of the carboxylate.
Oh, so I'm thinking if it were 3, if pH equals,
Ah,
because I'm looking
okay.
at the pKa.
So in between, okay.
So I was just saying
Okay.
the pKa.
Oh, if the pK were 3. Okay.
I'm confused about pH versus pKa.
Okay.
So
Okay. All right. So yeah, those are a couple of different things to review. So first of all, suppose the pH is 3. Well, we have to decide whether this is a low pH or a high pH, and the benchmark for that is the pKa. Well, what's the pKa for this nitrogen?
2.0,
For the nitrogen?
10.6.
So for the nitrogen, does this count like a low pH?
Yeah.
Yeah. So should the nitrogen have an extra proton here?
Yeah.
Yeah. So this would be the correct form of the nitrogen at this pH. Now the cutoff, what's the cutoff pKa for the carboxyl group for proline?
2.
Compared to this cutoff, is this a high pH or a low pH? High pH. So would we expect this to have a proton or lose a proton? Lose a proton.
So do we always get the zwitterion when you have a pH that's in between?
That's in between these two pKa's. That will always happen for the simple amino acids. For the more complicated amino acids, things will be more complicated, so we'll have to get to that later.
Yeah, when side chains.
That's
happen.
right. All the amino acids we've dealt with so far have been relatively simple because they didn't have acidic or basic side chains. So when we get to the acidic or basic side chains, we'll have to deal with that separately.
Okay, yeah.
However, before we do that, maybe we should try to clear up the difference between pH and pKa. Who has a pH? When we talk about the pH, it's the pH of whom?
Solution?
That's a very good answer. It's the pH of the solution. If the pH is low, what does that tell you about the solution?
It's very acidic.
That's right. And what does that tell you about the solution, however?
There's a lot of protons.
That's right.
So a lot of protons that will attach to those like nucleophiles.
There's a lot of protons in the solution, and therefore they have the potential to attach to any molecules that you put into the solution. That's right.
Protonate things. So
That's right. So you would expect this as a solution that's good at protonating things. So at a very low pH, you would expect that the solution is going to fully protonate any species that you add in. Now let's compare that to a low pKa. Well, first of all, we said the pH is the pH of the solution. A pKa is a pKa of whom?
The molecule.
That's right. That's an excellent. All right. Well, actually, it looks like you're already pretty clear in your mind about the difference between them. Most people don't understand that. Most people don't understand that the pH refers to the solution and the pKa refers to the molecule or the acid or the substrate. So when we say we have a low pKa, we mean we have an acidic molecule. This is something you might not have realized, that the word acidic has two different meanings. You can say a solution is acidic, or you can say a molecule is acidic. And even though those are similar, those are two different things. An acidic solution is a solution that has many protons floating around in it. An acidic molecule is different. An acidic molecule is a molecule that wants to lose a proton. An acidic molecule is a molecule that wants to lose a proton. So the word acidic is used in two similar but slightly different ways to describe either solutions or molecules.